I welcome you to this video on phototransduction. Phototransduction is a series of biochemical reactions which are initiated when light strikes the photoreceptor cells of retina. These light signals are converted into electrical impulses which are transmitted through the optic nerve to the brain for image interpretation. Thus, phototransduction is a signaling pathway that enables us to perceive objects in the presence of light. Since the rod and cone cells of retina play critical roles in this process, let us discuss the essential features of these cells in the next slide. So photoreceptor cells are very important. They are of two types, rod cells and cone cells. They have distinct regions such as outer segment, inner segment, cell body and synaptic body. So they are kind of neuronal cells. The outer segment of rod cells is filled with a photoreceptor called rhodopsin, whereas cone cells outer segment has a pigment called photopsin or iodopsin. The rod cells are responsible for blurred image or scotopic vision, night vision, and if there is any defect in these cells, it will lead to condition called night blindness. Whereas the cone cells are responsible for high acuity vision, which is color vision, sharp vision, or light vision. If there is any defect in these cells, you will see a condition called color blindness. So basically, in order to understand phototransduction in both cells, we have to understand the spectral properties of these cells in the next slide. It is important to understand spectral properties of these cells. For example, cone cells of three types. S cone cells absorb maximum light in blue region. M cone absorbs maximum light in green region, whereas red cone cells absorb maximum light in red region, whereas the rod cells lambda max lies between 400 to 600 nanometer. The other distinction is in the number of these cells. Rod cells are numerous, they are much higher in number as compared to cone cells in human retina. Why do the photoreceptor cells exhibit different spectral properties? It is due to variation in the photopigment present in these cells. Photopigment is a conjugated protein which is made up of an opsin protein part and an 11-cis retinal chromophore which absorbs light. The primary structure of opsins present in these cells is different, that makes them to exhibit different spectral properties. The genes for opsin in M and L cells are located on the X chromosome. That is why males are more susceptible to develop red-green color blindness. The other point to note is that 11-cis retinal is derived from vitamin A, and therefore vitamin A deficiency is tied with vision problem in humans. So in summary, although photoreceptor cells differ in their spectral properties, the phototransduction mechanism is fundamentally the same across them. Therefore, I will explain phototransduction in rod cells. The pathway begins with the absorption of light, which causes isomerization of 11-cis retinal to all trans retinal. So let us discuss the mechanism as it happens in rod cells. This is rod cells here. There is rhodopsin attached with chromophore 11 cis retinal here with the shown with the green arrow. The second component is transducin, a trimeric G protein having alpha, beta and gamma subunits. And the third one is phosphodiesterase or PDE enzyme having attached with its inhibitory subunit. So under this condition, all these three components are in their inactive state, similar to darkness or the night. When light falls on the rhodopsin, the chromophore is converted into all transretinal, which causes conformational change in the protein, making rhodopsin converted to meta-rhodopsin 2, which activate G protein, which with its alpha subunit, with the help of subunit, knock down the inhibitory subunit from PDE or phosphodiesterase enzyme, and thereby activating the PDE. So active PDE hydrolyzes cyclic GMP. As a result of it, the cyclic GMP concentration is decreased, which blocks the cyclic GMP operated calcium and sodium channels. As a result of it, very little or no sodium or calcium ion enters the cell, leading to hyperpolarization of the membrane, which inhibits the voltage operated calcium channel in the synaptic region. Calcium ion concentration therefore is decreased significantly, leading to inhibition of glutamate release. This is a neurotransmitter, and this disinhibit the bipolar cells, which activate ganglionic cells, and the electrical signal or impulse is sent to brain through the optic nerve for image interpretation. In the meantime, from romatorhodopsin, the all transretinal detaches from the opsin protein part, and therefore to reset the reaction, all transretinal must be converted into 11 cis retinal, and this happens in the retinal pigment epithelium through a pathway known as visual cycle, which we will discuss in a separate video later. Thank you very much for watching this video. I request you to like it, share it, and subscribe it. Until next time, you take care. Bye bye.